Everybody's always thought about going back in time, right? Like, you've dreamed about seeing something in the past that is no longer accessible. For me, some of my top things would be to see the origins, right? To just see what happened. That would be probably number one. If we could see maybe that first mitochondria when the bacteria came to live inside of another cell, like as a biologist, that's what I geek out. And maybe the first chloroplast, that'd be even better than the mitochondria because it's a plant. Maybe more recent things like the asteroid that hit off the coast of the Mexican Yucatan Peninsula and see what really went down at that time point 65 million years ago. Part of our own lineage, which I'm always curious about, there was a time only a couple tens of thousands of years ago when there was like 7 different species in the genus Homo that coexisted in the same parts of Western Europe. Were they friends? Were they not friends? How did that all go down? We now know that most people with European ancestry have 1% Neanderthal DNA. How did that actually happen? And I'm curious. Time travel, it's of interest to, I think, most people. Yet we often only get little snapshots, like little glimpses of it, like just a window into it. We can see it from the rings of trees, for example. I now know that you can go back in time and see these pack rat middens, these little nests of pack rats, and put together some things about the climate and the vegetation. The ear bones of fish tell us a bit about the history of the oceans. But I'm going to tell you a story that I went on, a journey that I went on with a student of mine, Alex Rinkert, and I have to make sure it's very clear here from the beginning that this work only would have happened with Alex's inspiration. So he's my co-author on this. He couldn't be here today, but he brings a lot of the bird expertise, and I just provide some background on the plants and some laboratory space. I'm remiss to not thank the San Francisco chapter of the California Native Plant Society. I met 2 Susans and a Sophie and a couple people who aren't here who helped make this happen. And the energy around native plants that even these little groups bring is super inspirational. And part of the reason why I'm here today talking about plants is because as a 19, 20-year-old myself, I would go to as many of these meetings as I could find. And thank you guys for providing that platform, both present and past. Let's dive in here to time travel, and I'm going to try to do something that I haven't done before. I've had an opportunity to talk a little bit about this. I have some new results to show you guys, but I'd like to go both back in time, but also promise a window into the future, equally or maybe even better than going back into the past. I'll leave that to the end. There's a picture of Alex. Alex is at the Cal Academy, which I can almost throw a frisbee to from here, and he's sampling a bird's nest. Alex came to me as an employee of the San Francisco Bay Bird Observatory, which is centered out of a lot of different places, but their field station is near Alviso in the South Bay. And Alex said we were put together by a common friend that he would like to reconstruct the plants around the San Francisco Bay. And I said, you must be in the wrong place, because at the time we were working on endangered plants of serpentines near Morgan Hill on Coyote Ridge. We were working on endangered plants in the sandhills of the Santa Cruz Mountains near Bonnie Doon. And I said, I don't really know a lot about the plants of the San Francisco Bay. He said, well, we're going to use bird's nests to do it. I said, you're definitely in the wrong place now, because I can identify a blue jay and a mourning dove and a couple of other my favorite birds, but why are you here? And then he said, there are some ancient bird's nests, some 120-year-old bird's nests, and the only way we're going to be able to do it is if we can look at the DNA in the plants of the nests. I said, okay, now I'm trying to get it, because we've studied DNA in a lot of different things. We've studied DNA in the rarest pines in the world. That's the Torrey pine that lives here in California off the coast around La Jolla and on the Channel Islands. We've studied the DNA of a couple of mustards, the Erysimum terridifolium from the sandhills, and jewel flower, Structanthus from Coyote Ridge. We've extracted DNA from bird feathers to try to sex yellow-rumped warblers. It turns out it's pretty hard to tell boys from girls at certain times of the year. So we can get DNA out of a lot of things. Most recently, we got interested in the guts of endurance athletes, because I had some students who were on the cross-country team, and they wanted to know what was growing in their guts, and was it any better than the people who weren't running 50, 60, 70 miles a week. So we could get DNA out of a lot of things. But I've never tried to get DNA out of dead plants, especially plants that are like 100+ years old. So I told Alex it was pretty risky. But he said that there is a confluence of natural opportunities for us to investigate a unique habitat of the San Francisco Bay. And here you can see, okay, we are in the San Francisco Bay Estuary. We are looking north from the South Bay around Bear Island off 101. You guys, you're driving on Highway 101 north, and all of a sudden the San Francisco Bay seems like it's right on the shoulder of the road. That's Bear Island, and we're looking at the San Mateo Hayward Bridge and maybe San Bruno Mountain in the background, and right now we're just over here to the left.
Looking north.
Looking north, thank you, sorry. We're going to talk a little bit about habitats that have disappeared almost entirely. We're going to try to reconstruct what they may have looked like using birds' nests by taking out their DNA, and then try to identify those plants, and I'd like to celebrate a lot of the plant diversity that we found. But I need to justify it with some background on the San Francisco Bay and a little history. So around the 1800s, everything in green here was considered estuary or wetlands, particularly wetlands. We're thinking like pickleweed, saltgrass, maybe some dodder sprinkled in there, the really wet stuff. That was in the 1800s, presently indicated in green, which is hard to see because there's very little left. We are almost completely out of wetlands. Whether you're in the North Bay here, San Pablo Bay, Sassoon Bay, or down here in the South Bay, almost all impacted, severely impacted.
Which was?
Yeah, I'm going to show you that. So in the mid-1800s, mom and pop salt ponds became popular, and they would levee off a portion of the bay, let some water in, dry it down, and then extract the salt. That's about all that I know about salt ponds. But it went from small, little, locally owned to industrial scale salt ponds. The ones that I remember flying out of like San Jose, San Francisco airport and looking down and just being amazed at what are these colors from outer space doing, and why are they so rectangular here on the edges of the San Francisco Bay? By the early 1900s, salt ponds were taking over, but we also see quite a bit of the wetlands lost to urbanization and human development. Does that answer your question? Mm-hmm. Okay, here we go. But I want to focus in not on the wetlands, because we know quite a bit of the plants of the wetlands. It turns out the actual plants that can survive in that really salty underwater environment are pretty limited. And then we know what's on the uplands, more or less, oak woodlands, valley grasslands, for example. But what's missing is this unique habitat called the transitional habitat. And the transitional habitat is higher than the wetlands, yet lower than the uplands. And to give you a sense of what it might have been like in the 1860s, here's a quote from somebody who grew up around Mountain View. His name was Beardsley, and it was written up in 1926, but he's remembering his childhood, where there was a great salt marsh with all its winding sloughs and creeks covered with sapphire grass, which was what he called it, and we now know he was referring to Salicornia. Tufts of Grandelia. Next was a line of natural salt pan, and next again was a strip of land of varying width. This is the transitional habitat, with from a few hundred yards to a quarter mile wide, with short, wiry, hard grass. That's Disticus spicata, most likely. This was a first-hand account of the wetland becoming uplands and the transitional habitat therein. Unfortunately, that transitional habitat, which used to be, like he said, up to a quarter mile or larger, I'll show you in a second, has been sometimes compressed to literally just a couple meters. Like the distance between one of the Susans and me is the entire transitional habitat that should extend forever because of the levees that have been introduced in order to save urban environments. We are above the level of the high tide, but not necessarily above the level of the king tide. So this is the current status of the transitional habitat at Bear Island. You'll see it's quite compressed into just a few meters here between the high tide line and the king tide line. So very rare, but occasional inundation. And here is the historical perspective from that same 1926 paper. He was interviewing some people from Malviso who were recounting their memories of the area. And some of these regions in Mountain View, this is Highway 101 superimposed in blue here, show this transitional habitat going from relatively thin to quite wide and expansive. What I'm trying to emphasize here is this isn't just a little strip on a levee like what it looks like now. It was a habitat, and it had a unique plant community, the same way the redwood forest or a valley grassland or a oak woodland has a unique community. This was a unique community transitioning from the wetland to the uplands. It has a lot of important functions in addition to being a home to a lot of plants that we all are here to love. For example, it provides buffering during a lot of flooding, both natural and introduced by humans. So it would provide a buffer for sea level rise. It will allow us to adapt to climate change if we have these longer stretches that has native vegetation rather than a short levee that could collapse. And although we're here for plants, we know that things with backbones that are a little bit charismatic can sometimes get the attention and the conservation protection that helps the plants out. So Ridgway's rail is a listed endangered bird that uses the transitional habitat. It's distinctive because of its black and white stripes on its haunches here, skinny legs, pretty wide toes, allows it to move around on the mud, but at the same time feed from the transitional habitat. In addition to Ridgway's whale, we have a little tiny mouse, this salt marsh harvest mouse, another listed endangered species that requires the transitional habitat as a refuge. That's pickleweed, and I don't know if you know pickleweed or have seen it recently, but it's a pretty small plant. And there it is with the salt marsh harvest mouse, weighing in at 1/2 ounce and 6 inches long. Half of that is its tail. So it's an adorable little thing. But imagine you are 1/2 ounce and 3-inch body, and you're living in this habitat. During a normal high tide, you have a lot of habitat in the wetland you could occupy, some salicornia, that's pickleweed, maybe some saltgrass, and you might even use a little bit of the lower edges of the transitional habitat. But during the king tides, these salt marsh harvest mice need a refuge. And the refuge suddenly shrinks. But the transitional habitat is its guaranteed home, right? If it gets stuck on this island, the chances that it will successfully swim to the transitional habitat is lower than if it has a larger transitional habitat to occupy in the first place. So people who protect vertebrates are interested in these things, and that might be one way we can motivate the preservation or the recreation of lost transitional habitat. By the way, if you have questions or want to see something, holler. This is a story, and I'm glad to answer questions as we go. Yes?
So that the mouse, does it swim?
Yes.
Does it have any special properties that gets rid of the salt for it?
I don't know the answer to that. That's a good question. I know that the Ridgway's rail is actually famous for having a unique salt gland that allows it to drink salt water and then excrete it. The salt marsh harvest mouse I'm less familiar with. And honestly, this is about as close as I've ever been to one. I'm sure I've been closer, but I didn't know it. Good questions, though. Exactly. Okay, the transitional habitat. So the earlier map was of wetlands, and it was almost all gone, 90% missing. This is the transitional habitat, and the areas in gold are all thought to be candidates for restoration. A really heavy amount here on the peninsula and into the South Bay and the eastern portions of the South Bay, and then you'll see some stuff here in San Pablo Bay and a little bit in Sassoon Bay. A lot of people are interested in doing this, both by choice, but some of them by force through mitigation requirements. However, this restoration assumes that you know what to restore. And that part, Alex came to me and said, I don't think we know. I'm working for this organization, and we're working on restorations, and it's unclear exactly how we know which species. I thought, well, this will be easy. We'll just go to the Cal Academy or UC Berkeley to the herbarium where all the dead plants are kept. And we'll just look up the oldest plants from this area And it'll just tell us, you know, from the 1700s, early 1800s, it'll say transitional habitat, it'll say Alviso, maybe there'll be some GPS coordinates, right? Yeah, that was my naivete. It turns out we know virtually nothing from plant explorers. If there is information, it frequently just says San Francisco Bay. More likely it says Northern California, and sometimes it just says Northern Mexico, because it's that old. So we have very few extant reference sites, very few herbarium specimens to work with, and nobody, unfortunately, took really thorough notes on the transitional habitat. Lots of information about the wetland, but it's mostly pickleweed, and lots of information about the oak woodlands. But this little transitional habitat wasn't acknowledged as a really important unique community until long after it had been largely lost, except maybe preserved in a couple of birds' nests. So let me tell you about these historical description really quick. I quoted from this Cooper paper, who interviewed some scientists from, some residents of Alviso, and from that information they came up with a list of what they thought were some of the key components of the transitional habitat. This is a little plant quiz because I feel like I'm in good company here. We've got the following plants. There's going to be 10 species, and they're all secondhand information. Keep that in mind. Amazonia congesta, looks like that. Centromatia, Symphyotrichium. You said it's a theme here, if you recognize any of these names. If not, I'll spoil it here in a second. Another Symphyotrichium, Baccharis, Helianthus, Iva, Solidago, Heliotropium, and Rose. If we planted only these plants as the transitional habitat, it would be a beautifully colorful summertime, because a lot of these are Asteraceae and many of them are tar plants, Amazonia and Centromatia in particular. It would be mostly annuals, except for the rose and the coyote brush. That's Baccharis and Rosa. But we would be planting 8 out of 10 sunflowers, and it seems like this is a biased outlook. That particularly, it would come up, this person was probably interviewed in the middle or late summer when most of these things are in flower. So this is the first-hand knowledge that we have from around the 1860s, 1870s, then published in 1926. Could we use some other source of information to inform what to plant there? And Alex came to me with this idea of bird's nests, and I said, well, why did anyone collect bird's nests? Like, was there any money in this? We're talking about bird's nests in the 1880s, 1890s. It turns out that the bird's nests were bycatch. People were out for feathers, first of all, which were worth their weight in gold for women's hats in post-Victorian times in the East Coast of the United States, and they even sent to Europe. But they were also interested in eggs, and eggs were amazingly valuable for collectors of bird eggs who had lots of money and wanted many bird eggs as they could find. So these people, naturalists, would go out and collect eggs, and in order to find eggs, they would come across a nest. And then they would have the nest, and some of them had the wherewithal to then donate the nest to the museum. So the eggs went and fed them, and the nests went to the museums, where I'm pretty sure many of them have sat since the turn of the 1800s to the 1900s, until Alex and I went there. For example, here's a bird's nest collected by Joseph Grinnell. And I think of Joseph Grinnell as the Alice Eastwood of animals, or the Jepson of vertebrates. So he was the first director of the UC Vertebrate Museum. He is a famous bird biologist and vertebrate biologist. And I think anyone who knows about vertebrates or zoologists would probably be offended if I analogize him to Alice Eastwood or Willis Lynn Jepson. But those are my superheroes, and I'm understanding that this is an important person. He's collecting around 1915. Grizzly Island, a lot of detailed information for that age nest. At this point, that's about a 110, 111-year-old bird's nest. It has a record of how many people have gone in to look at it. In the first few years, a couple people looked at it. Nobody looked at it since, until Alex went there. and got permission to take a couple small pieces out of the nest for DNA. Again, these are very important historical collections, and they probably are harboring all sorts of information about the past that I haven't even begun to fathom. But we know the nests are made out of plants.
Even dead bugs, huh?
Probably. I have a story about insects near the end, I promise. Remind me if I don't tell you. Alex counted them, and it's not just one or two of these. There are hundreds of nests, and I think they have enough detail to get us to the transitional habitat of the edge of the San Francisco Bay. And they go back, we're now working on about 150 years, to the 1870s. This is the frequency of birds' nests, and these are in bins of 10 years. They are not all at Cal Academy or at the UC Museum of Vertebrate Zoology, but they are scattered around the country. But this is what they look like. I mean, I was hoping that we could just stop at the nests, look at the plants. They would have all the most important traits, like a grass would have the glooms, and it would have the awns, and it would have the auricles that clasp to the stem, and it would tell us, it would be screaming out, yes, this is an elamus, or no, it's a lamus, or some microstachys. But it turns out the nest is beautifully arranged like that, and we're working with fragments that on a good day look like this, which is maybe a piece of grass here, maybe something with some ridges and knobs here, relatively nondescript, and not enough to restore the transitional habitat. But we don't want to take apart the entire nest. We want to just sample small pieces. Oh, so we're going to try to get some DNA out of there. We're going to go from the nest to a DNA extraction. The DNA extraction is going to tell us a little bit about using some techniques, biochemistry techniques, the name of the plant, and hopefully we could then use that to inform restoration. Are you ready? Okay, here we go. The question of the moment is, can we use birds' nests to identify their DNA? And then we can tell a little bit about what makes these nests work or don't work. But I need to first highlight the birds, because these are where the nests come from. And we can't just pick any bird's nest. We need to focus on a bird that makes their nest from the transitional habitat, and hopefully only the transitional habitat. We don't want a bird that goes up into the oak woodland, collects their nest materials, and then brings it back, because then we would be reconstructing the transitional habitat from the wrong habitat. And I thought, well, this is impossible, because I just see birds all over the place all the time, right? Alex points out that this is not true of all birds. There are a couple birds in particular that are extremely faithful to this transitional habitat. And to prove it, Alex went out and, first of all, he found savanna and song sparrow nests. He then found birds actively making that nest. So to find the nest is relatively rare. To find birds actively making the nest, it seems even rarer to me. And then, with the patience of only a birder, he then watched the birds travel out from the nest, estimated the distance that they traveled before they came back to the nest. He never lost sight of them, which seemed impossible to me. But remember, we are in a relatively open visibility habitat. And on average, he found 11 of these birds moving on average about 17 meters, and it's relatively tight, between 10 and 20 meters. So they tended to move laterally along the edge rather than 10 meters into the wetland or 10 meters away from the wetland into the upland. And that was very encouraging, plus all the additional information that people who know the savanna and this particular subspecies of song sparrow, that these birds are very specific to their habitat. So this is this interesting confluence between a bird that's faithful to its habitat, the availability of old nests in a habitat that needs information. We are going to start with a pilot study of 2 nests from 1915, including that Grinnell nest. One of them's over here from the Oakland area, and the other one is from the Sassoon Bay in the East Bay. And then we've got 2 contemporary nests. These are kind of our controls just to compare. From 2003 and 2017, those are both from the South Bay. This is Alviso, where the San Francisco Bay Bird Observatory has their field station, and this is from Bear Island. And these nests were studied systematically. They are very specific architectures. They're unbelievable builders. They usually use rough, coarse material to provide sort of a foundation for the nest. Often includes some woody debris like woody twigs or larger, heavier plants. And then as you work your way from the outer structure into the central lining, you get finer and finer materials until you get these really thin, fine annual grasses where the eggs will be deposited and grown. We're working with pieces that are like 1 to 2 centimeters. It had to be a contiguous piece of material, so we only had one plant on there. Turns out later we'll learn why that's not necessarily true, because DNA is everywhere all the time, but we thought we were getting just one plant. A centimeter is like the width of your pinky, more or less, so we're talking about pieces of grass that are like this big or this big. Pretty small bits to keep the nest intact. If you're curious about how we do this DNA work, we use a kit, and it comes with these little columns, and we'll take the pieces of DNA and we'll put some balls in there and shake it up really hard using an instrument called a bead beater that homogenizes it. It makes it a little bird nest milkshake, and then we'll use some buffers to clean it up, bind it to the column, a couple of washes, and at the end we'll have just a couple drops of liquid that we think has some bird nest DNA in it. Yeah, question?
Were you able to find bird DNA in it?
Yeah, we have, the next step is to use some little molecular tweezers. We're gonna go in and look for a really specific chunk that's gonna tell us what species it is, because not every piece of DNA is equally useful. With those molecular tweezers, they are organism specific. So we need plant tweezers to get plant portion out, which is what we focused on. We now have insect tweezers that can go in and pull insects out of these liquids. And we've also verified a couple of the nests for the bird to confirm that it's the right species. Not from the ancient ones, only from contemporary nests. So we're gonna go in and try to pull out a really small region and amplify it, because it's gonna be highly degraded in order to do the sequencing. Good question. The amplification process is called polymerase chain reaction, and it's where we go from a couple pieces of degraded DNA to thousands or hundreds of thousands, maybe even millions of pieces of one small region, these little tweezers that went in for a specific spot. Because our method is only as good as we have a reference panel to compare the sequence to of known plants. We use agarose gel electrophoresis to separate out the brands, and then we do some sequencing. At the time, we were using a company called Sequitech, which ironically was right off Highway 101 in an area that was probably impacted by the transitional habitat. Just like the transitional habitat, Sequitech is now also extinct. Sadly, because I had some friends who worked there. We then use computers to help analyze the data, and the initial data sets were relatively small, and we could do it on our own computers. Later I'll show you some larger data sets that require some more bioinformatics heavy lifting. But we need this reference panel. We need a group of plants that we think might be there, broadly defined. So Alex and I and some experts came up with almost 100 species that we said, these things could potentially be here. Nobody knows for sure. It included the ones that the initial paper from 1926 suggested, as well as another 90 or so of them. And almost all of them had this region called the internal transcribe spacer, or ITS. That's what we're going to grab out with our tweezers in GenBank. And GenBank is a free open access database of all previously published DNA sequences, including the human genome, the mouse genome, most bird genomes, as well as every plant DNA sequence that's ever been published. And everybody has access to it. The cool thing is that almost every species that we thought could be in the transitional habitat was in there, so that's a good start. And any species that wasn't in there, that remaining 9%, there was a close relative that was in there. Typically, the close relative doesn't live near the San Francisco Bay, so by inference, you could say, well, it's probably, say, a rose, but it's not that rose, because that rose is in Oregon, so it must be a California rose. More on that in a few minutes. So we have a nice reference library. We're going to use a tool called the blast search to identify the best matches. That's an algorithm for identifying DNA sequence matches. And then we're going to measure some genetic identity, like how similar is our sequence to that. And finally, we're going to build some trees. These are phylogenetic trees, like a family tree of these samples to see how they're related. And we're going to look for who is the closest relative to our bird nest sample. We're looking for the closest relative, like the sister. Here's some initial results. These are some of the plants that we found from these nests. The first one is from the ancient nest, we'll call it ancient, 1915, from Joseph Grinnell. That's rose. And the second and third, all the rest are from the contemporary nest. This was our first pass. We had some, I'm going to tell you about these individually. We had about 60 nest samples taken from those 4 nests, about 15 samples per nest, and only 13 of them worked on our first pass, which was a little bit of a bummer, 25%. But keep in mind, half of those were over 100 years old. I was just amazed that even one of those ones worked. It turns out of those 13, more than half of them are not even land plants. So there's a bunch of green algae in there, and there's even a ciliate, which is a protozoan. It's kind of a quasi-animal, depending on who you ask. And I can talk more about those if you're interested, but I was sure those were all contamination from my lab, but we have these negative controls where we do everything, but instead of with a bird nest piece, with water. And we never got any green algae in our negative controls. We never got any ciliates in there. We never got anything in there because it was just water the whole time. Turns out all those green algae are different species and they all live in saltwater conditions. So it's likely part of the nest, and I assume it's part of the nest that got inundated during a king tide between the time that the bird used it and the time it was collected. The ciliate is also described as a saltwater endemic protozoa. I can tell you more about those, but I'm going to focus on the plants because that's what I'm here for and that's what we're trying to restore. The rose is what worked from the old nest, and we got 5 from the contemporary nest, but each one tells a slightly different story. Let's go. The California rose, the ITS sequence says that's definitely it. It's a perfect match. And the Cooper reference, it was in that list from the 1926 paper. That was encouraging. When we pull down the sequences here, you'll see a bunch of other roses. These roses are more distantly related. Some of them are from the East Coast, some are from Europe. Here's our bird's nest sample, and here's our Rosa californica. And then there's a statistic here. We're looking for 70% or above to be statistically sure, but we're close. We're not quite there. We can know it's definitely not any of these other species. However, there's a couple local roses that aren't in our database in this case. For example, Rosa nuitcensis. And having grown up identifying plants around here, I remember that being from around here, but also for being more common in higher elevation positions. And so we excluded that one based on just natural history alone. Otherwise, we're pretty sure it's Rosa californica. We also use iNaturalist to help out, or we also use Cal Flora. And you can see there's 4 records of that Nucana rose in the San Francisco Bay, although they're usually a little bit more higher elevation. Meanwhile, there's over 120 records. And I did this update this morning, and it's like 10 records in a small area of the San Francisco Bay versus 2,000 records in that same square, meaning that there's like a 200 times higher chance that it's Rosa californica than Rosa nucana. To confirm it, we would sample Rosa nucana, add its DNA to the tree, and make sure that it's not the sister. This is what it looked like, which in retrospect is, yeah, sure, that's a leaflet of the pinnately compound rose leaf. Do you see the green bits in there? They're a little bit etched on the edges. I mean, in retrospect, you know, it's a 100% match, but if you asked me this ahead of time, I'd say, I have no idea what that is. The fact that they were still green is pretty amazing, and that probably worked in our favor. That means that the plant was collected and then dried out really quickly, like a good herbarium specimen, and didn't go through a long period of slow death. That's from the old nest of Joseph Grinnell. I'll tell you another story here about small fescue. This is from a contemporary nest from only about 20 years ago. It's from the, maybe 15 years ago now. It's from the Bear Island area off Highway 101. And it's a match for the ITS. It's not on Cooper's list because he had no grasses. You remember, mostly sunflowers. And when we put it into this tree, it pairs with Festuca microstachys, this slender small fescue with a 99% bootstrap, meaning it's almost guaranteed to be that, as highly confident as we can be. And it's probably not any of these other fescues. The cool part of this story is that this was from a restoration site when in 2015 was completely bare. The Botanical Survey from that area before they cleared it out didn't have any Festuca microstachys. However, part of the restoration protocol was to put in some grasses, and they picked Festuca microstachys. Then, subsequent to that, Alex found the nest, and sure enough, it had Festuca microstachys in it, suggesting that not only are we identifying the plants that the birds are, that are potentially in the transitional habitat, but that the birds are actually using the plants restored. It's particularly good because it's a very thin, soft, fragile fescue, and it makes for perfect spots for the lining of the nest. So we think the birds are actually using the plants in the restoration, at least some of them. We have more Elaemus or Elmus. This is probably Triticoides. It was a little bit of a tangle. I don't know if anybody is interested in these grasses, but the names keep changing between the two genera, and the samples landed in multiple places. They seem to always cluster in or around this Triticoides, which is a native California grass, even though it's a site that's filled with all sorts of non-native plants. So that was exciting to see. And another grass to add to our list of plants that could be planted in the transitional habitat. However, like, if you tried to convince me that this is a grass, let alone a Elaemus Triticoides, you know, it has this unique clasping auricles where the blade comes off of the culm, and yeah, the bird didn't pick the traits that I need to identify it when it made the nest. What was it thinking? Alas, these small bits of leaf is what we're working with, and sure enough, I think they are definitely natives. We also found some non-natives, not surprisingly, and in fact, this all came from this nest here. This is a song sparrow nest. We're now working with the contemporary nest, and you'll see a picture of the bird there. The nest is unbelievably designed. I'd like to pass this around as long as you take good care of it. It's got some heavy architecture on the outside for the stability, and then some really fine materials on the inside for where you're going to leave the eggs. And as you look through it, see if you can't make out some of the siliques. That's the fruiting portion of some mustards of Cardamine hirsuta. You might know this plant as one of the first weeds that show up in my yard right around January or February. And after its little white flowers show up, you can kind of brush it with your hand, and it will explosively set its seeds in front of you. It wasn't until after we did all this work that I looked carefully at it and I said, oh, of course, those are the siliques of a Cardamine. Yeah, that comes from the San Francisco Bay Bird Observatory, and it wasn't on Cooper's list. It's very highly likely Cardamine hirsuta with a strong bootstrap. As a control, I was expecting to see some pickleweed DNA in there, and gladly, of the 6 plants that we got, one of them was pickleweed. It turns out which pickleweed is a little bit more complicated, but I think that's mostly a complication of pickleweeds rather than a specific problem with the San Francisco Bay wetland. The bootstrap is relatively high for our sample, being close to one of 2 different pickleweeds. One's called Virginica, sometimes called Pacifica. Sometimes Pacifica has a north and a south race to it. The other one is Salicornia perennis. It seemed like that one is less common in the San Francisco Bay, particularly in the region where that nest is from. Someone once said that pickleweed taxonomy, like the names of pickleweed, is like a taxonomic nightmare come true. I'm thinking it's a northern variant of Salicornia pacifica, but I'm always attentive to the possibility that 100 or 130 years ago, when we start looking at more ancient nests, that there may be something there that's different, or maybe not here anymore, or maybe completely extinct. And that's one thing that drives me and keeps me up at night and wakes me up in the morning, is to think that there may be an undescribed species, or at least something largely out of its way. So I pay really careful attention to this. Otherwise, I just say, yes, it's pickleweed, let's move on. But I wanted to make sure we got the right one, and so I'm pretty sure it's most likely going to be Salicornia pacifica, which is more of a positive control than any kind of discovery. Okay, we're going to go to the next level, because we didn't have very many nests. Only a quarter of them worked. So our sensitivity and our success rate was pretty low. And some of them we were not 100% sure what species it was. So we got 20 old nests now, and I'll show you the distribution, but they're almost all over 100 years old. And the great thing about giving this talk is that every year that passes, they get older. 75% of them are the song sparrow and 25% of the Savannah sparrow. I love the birds, but it turns out the nests are composed of almost identical components regardless of which species, which is encouraging. It suggests that both birds are doing the same thing, hopefully being true to that habitat. And that meant Alex and I were traveling all over the country to these museums. I want to point out a little known museum. This is the Western Foundation of Vertebrate Zoology. It's in Camarillo, down by Ventura. They have the world's largest collection of bird nests. And it's a place that's struggling for funding and has very few visitors, and they were so excited to see us. As you can imagine, the world's largest collection of birds' nests is rarely accessed, but we may have given them some justification. These are some of the other places that generously allowed us to take some samples, including just down the way. We're going to do something slightly different. We're going to use next generation sequencing. It's going to rely on a different tool, which is about the size of a harmonica. That other machine to do the previous work took up a large portion of the laboratory. It's a special machine, and I had to hire out a different company. This one, my students can do as part of a class on these little devices right here. It uses microfluidics, which is really, really small amounts of liquid to flow DNA through a small pore, and doing so, it changes the voltage. And the A's and the T's and the G's and the C's, those are the DNA base pairs, have slightly different voltage when you send electricity through them. So it gives you a readout of the bases, and you can read them relatively efficiently. This is the thing that promises like your genome for less than $100. I think it's closer to $10 or $1 now for your genome that you could get by paying or through a healthcare provider. But these are safe, just don't.
Full sequence?
This, we are still amplifying this little chunk with our tweezers. We're just putting those amplification products on this, so we don't need to sequence the whole genome of the plant, only the little piece that we're after. Ironically, one of the steps in preparing it is to cut the DNA into known fragments, but the DNA has already largely degraded. We put on some known linkers there, and we're hoping for a higher success rate. That's as far as I want to go. I'm glad to talk more about the technology there, but it's really just the means to an end for me, because I really want to get to the plants. It does some fascinating things called bridge PCR in there, and makes some clusters with pretty colors. These are the colors of the A's, T's, G's, and C's. Again, more than glad to talk about that, but let's get on with the plants here. One thing that this can do that the other technique couldn't is that we needed to have just one plant in our tube in order for the other technique to work. It needed to be homogeneous. However, this technology allows us to have multiple different types of DNA in one tube, and it can sequence them separately and send that information to us.
Wow.
So here's some results. So again, we have 20 nests, and they are all over 100 years old, and so no more contemporary stuff. And I've divided the results into the Savannah sparrow and the song sparrow results, and into some basic categories here. You remember that previously we had some grasses and a couple herbs with a shrub. We now see some grasses, some herbs. Collectively, they explain more than 3/4 of the transitional habitat, a couple shrubs, and the evidence of a few trees. And the differences between those two birds, like I said, are pretty similar. But which plants are they? Let me tell you a little bit about the years and then I'll show you the plants. This is whether they are native or not. We had only a few nests from 1875 and before, and you'll see 100% of the contents of that were native. The chances of going to any area around the edge of the San Francisco Bay and picking 20 or 30 plants and getting 100% native is about 0, right? There are very few places you can do that anywhere. It drops down to around 75, 80% in the first 15 years and then levels off. So it seemed like there, at least for what the birds are using, was an initial invasion of a couple non-natives and then a leveling off. Now we're not actually seeing the habitat, we're seeing the birds' use of the habitat, so I need to make sure it's qualified in that way. There was a couple of years here where we didn't have any nest materials, but I want to emphasize that it's pretty leveled off there. Let me tell you a little bit about some of the plants that we found, and then equally interesting are some of the plants that we're missing. But let's start with some of the natives that were all. Here's our Salicornia again, and Disticus spicata. These are probably from more on the wetland side of things. Spartina, also wet feet on Spartina all the time. But we found some shrubs like California sagebrush. There's that grass, Elymus triticioides, that we had from before. And some famously salt-loving plants like Jaumia. It's succulent, it makes a beautiful little sunflower and is commonly found in these habitats. It suggests that whatever we were doing seemed to be picking up plants from the right area in the world. We got some non-natives too. You'll recognize these spiraling legumes of the Medicago polymorpha related to alfalfa. Some annual grasses. This little linum, which I'm still trying to put together. It's definitely not the plant species you use and distantly related for the one used for linen, but it's in the same genus, but definitely not that plant. It's a common ornamental from gardens, which people would have been gardening in the late 1800s around the edge of the San Francisco Bay, or whether it has some other identity I'm still working on. And some radishes. It turns out the mustards are going to be overly represented because they're mustards and they do great here in California. However, we picked up some unusual things. Again, high sensitivity. And I thought, well, this looks like what the people in the lab, the students, were probably eating for breakfast and lunch. They tend to eat much healthier than I do, and it looks like a very plant-friendly diet. It turns out our negative controls for the DNA extraction and for the PCR, and then all the way to the sequencing, and we spent extra money just to sequence water, confirmed that none of these things show up in the negative controls. So this is not in the tubes, it's not in the tips, it's not in the water in the lab. However, as I started looking into it, I realized some of these things could be agriculturally important from the late 1800s, early 1900s. For example, in California, I think you find tomato DNA is probably just about everywhere, and citrus is available, as are things in the genus Prunus. A huge diversity of things, but I think that one's an almond. And some of these things, probably not. I don't think you can grow sesame seeds and bananas very successfully around the edge of the San Francisco Bay because a little frost will do them in. However, the San Francisco Bay was probably not treated with the respect it deserved, and I bet a lot of waste went into the transitional habitat. And these are the types of things that you might pick up as food waste around the edge of the San Francisco Bay. I'm clarifying, we don't want to plant sesame, bananas, and cantaloupe around the transitional habitat. But I think we are starting to get time travel, not just botanically for the native plants, but we're starting to see some cultural influences here, which is kind of exciting. Remember, we're going in with plant tweezers, so we're picking up mostly plants. We will exclude those from the final analysis, and we now have a list of about 71 plants. Warning, you're not supposed to read any of this. The point is, there are 71 plants that we've identified, and I want to walk you through some of the highlights. The two bars here are the Savannah sparrow and the song sparrow, and they go back and forth, and there's really no significant difference between the two in terms of their plants. The plant species are indicated here, and how common they are is the length of the bar, which may be proportional to how often they were in the nest, but there's also some caveats on that. But it was nice to see some things that we've seen before. The rose is in more than one old nest. The rose is in many old nests, and it's the same species of rose, Rosa californica. And it's relatively common. These bars are a little longer. But we saw some new things, and some things that I think are still a little unclear to me. For example, there was something in the genus Senecio, like a groundsel. It's not the weedy Senecio that we see in every street corner around here, but instead it either allies with something called Hydrophilus. That was encouraging, the water-loving Senecio, and I know that's around the edge of the San Francisco Bay, but I couldn't exclude Triangularis. And it's a bit of a mystery, so if anyone knows about Senecios around the edge of the bay, I'd love to talk to them more. Also, a bit of an unsure Solidago could grow in the higher edges of the transitional habitat. It might be Solidago spathulata, but it might be something else. We need to go build the reference collection a little bit more thoroughly before we can confirm that. And I mention these because these are the ones that I said keep me up at night and wake me up in the mornings because they didn't fit perfectly. These ones fit a little better, and I don't think they're transitional habitat, but I think they're more evidence that the San Francisco Bay drains water from a lot of places, some of them natural and some of them maybe not natural. We have pine DNA, relatively high amounts of pine DNA. We have willow DNA. We have a lot of oak DNA, some redwood DNA, definitely not the transitional habitat, and even some eucalyptus DNA. There's something that at least these top four all share in common. Pollination by wind. They make a lot of wind-borne pollen. So I bet a lot of this settles in the water and then collects and may have contaminated the nest. We call this bycatch. It's the bycatch of plants on a bird's nest.
Wow.
We are pretty sure now that birds are using this particular willow, and this eucalyptus tree is an interesting cultural moment because around the 1850s, I think, are the first records of eucalyptus in California, which turns out to be in Berkeley, where some of the first eucalyptus groves were planted. And then within the next couple decades, completely took off in California for lots of reasons determined to be relatively useless for what they wanted to use it for. Anyways, nest from 1850, eucalyptus DNA, it seems like the right timing. Here's some native shrubs that we found. There's that Artemisia californica, relatively common. Some Ceanothus thyrsifolia, that's the commonly planted one. I think that's the one you might have in your yard, for example. Baccharis, this is a different species of Baccharis. We have two species, the coyote brush Baccharis, but also this Baccharis glutinosa, which is more common in wet spots. And then, some salt-loving plants like saltbush, Atroplex. In addition to the things we found, there were things that I was just stunned to not find. For example, probably the most common plant in the world. My buddy studies grasses, and he studies grasses of the world. And he said, Justen, Avena fatua, Avena barbata, whatever you call it, is probably, like, if you count plants, there's probably more of that than any other plant in the world. Agreed? Not there. It was collected in the 1850s in Western North America, but it doesn't look like it was really settled in the San Francisco Bay until the late 1800s, and I'm not 100% sure it was in the estuary until like 1890s. And what we know is it probably wasn't permeating the place, or else we would have picked it up. It's in all of our contemporary nests, but it's not in any of these nests that are 100 years old, which is, bittersweet to me. It means, number one, it's a relatively new invasion, but number two, it means its velocity has been amazingly fast. A very, very expensive weed of the San Francisco Bay is Phragmites. Millions and millions of dollars are used to try to control it. It invades the wetland, and then it forms these mats. It then basically turns the wetland into dry habitat, which then collects a bunch of dead plant material, and it's a fire threat and a clogging threat.
What are the common names?
Yes. So this is oakgrass. Sorry, thank you. This is just a common oakgrass. And Phragmites, I only know as Phragmites. Sorry on that one, but it's like it's a monocot, grassy, reed-like thing. And if you go to any area around here, unless it's been controlled, it'll be omnipresent. But a colleague of mine in Santa Clara has told me that they have references that it's only 30, 40 years old, so it's impossible for it to have been in a nest from the 1880s, 1890s. These are nice internal controls. That's what Phragmites looks like, by the way, the middle one.
Also known as common, what are they, common reed?
Yeah, it's like a tall, grassy wetland thing. Brass buttons. I wish it was native. It's adorable. In fact, the people who preserve the wetlands for duck habitat don't care, and they're planting it. So it's a relatively new member of the San Francisco Bay, not in any of our nests, but is present in the bay now. All right, so we definitely have DNA coming out of these nests. It's telling us all sorts of stories, the interactions between birds, like in Festuca macrostachys. It works for 100-year-old material. We have some details about what nests work better than others, and I'm glad to talk about that later. But I promised to tell you a little bit about not only going back in the past, but maybe moving into the future. Do we have like 5 minutes to tell you about the future? Question?
I have a question about, I'm surprised that they're not picky at all, right? The birds, about what they use. I thought maybe with their beaks, they would need certain species that they would use more often than others, but it seems like, they'll use any old thing that's laying around.
Yeah, that's a good question. You would think that given their specialization to their habitat and their morphology, that they might only pick 1 or 2 plants and just do that. But we're picking up over 70 different plants from their nests. Careful, some of those are environmental DNA that's bycatch. But I think a lot of these are probably used by the bird. The bird's not just making nests in there, they're also living in there. So all sorts of plant DNA comes when you live in a nest in an enclosed space and you don't clean it very often. So some of these plants may be signs of their excrement, for example. Speaking of excrement, let me take you to the future, because what we want to do is learn about these plants, and then we want to restore these habitats into the future. And we started thinking about how to do that using the bird banding station over in Coyote Creek. This is Joey and Karina. They're 2 students who are either in my lab or recently graduated. One of them is starting at the University of San Francisco. Actually, I take it back. They're both starting at the University of San Francisco in different programs, biotechnology and immunology. Alas, don't blame them for that. They work with the San Francisco Bay Bird Observatory and our department to invent a field called molecular scatology. So scatology is the study of scat, and we are going to try to identify the plants used by birds from their scat, particularly to ask the question of whether or not restorations have been a success or not. We're going to be down here in the South Bay near Alviso at CCFS. That's the Coyote Creek Field Station. We have a couple sites at Jasper Ridge as a control where there's no active restoration. But this is Stanford's preserve. We're going to focus mostly on this result. And people have been restoring these for some time. We have a list of plants that are been restored at Coyote Creek Field Station, and what they wanted to know is, yes, the plants are there, but are they part of the ecosystem? Are they being used at different trophic levels by the birds or maybe insects that the birds are eating? So here's a couple of birds that we have seen and banded at the Coyote Creek Field Station, and you'll see this one eating some asters, and this one gorging on some elderberry, which will come up again repeatedly. And sometimes it's obvious. Unlike the bird's nests that were completely cryptic, sometimes it's obvious. These birds are captured in a mist net and then put into a small sanitized bag. The first thing they do when they go in the bag is they poop. And then the second thing they do, they take the bag to the field station where they're going to make some measurements, give it a nice little anklet if it hasn't been captured before, and then set it free. Is you look at it, and its whole head will be purple, and you'll have caught it right next to an elderberry, and not surprisingly, the DNA from the poop says Sambucus or elderberry. So we're not only going to look at what proportion are native and non-native, but if they are native, are these plants part of the restoration, or are they eating plants that aren't part of the restoration that maybe we should introduce to the restoration? The forward-thinking, predicting the future part. That's as close as we're going to get to predicting the future. We find a bunch of Wilson's warblers, we have spotted towhees, a lot of gold-crowned finches. I could talk more about the diversity of birds, but we now have over 100 samples. They reliably poop in the bag every time. The bag is sterilized in between bird catches because they don't want to transfer any diseases, so that worked well for our DNA work. We use the same DNA columns for extraction and a bunch of the same techniques. We can sequence them many ways, but the take-home lesson, you'll recognize this little tool here as the one that's passing, the two that are passing around. The take-home lesson is we're going to ask, is it native or not native? And if it's native, is it part of our restoration? Or should it be part of our restoration? It's just growing wild out there. Just be aware, the edge of Alviso is a weed patch. They've done a nice job with some restoration, but if you walked around Coyote Creek Field Station and the levees in the South Bay, 90 to 99% of the plants that you bump into are going to be weeds. So the expectation is that most of the bird poop is going to be weeds. That's like the null hypothesis. So we're going to divide it quickly into native versus non-native. Here we go. And the dark bars for overlap with the restoration and gray for no overlap. We had about 17 plants identified that were native. Most of those were part of the restoration. This is about 45% of all of the plants that we've studied. There were 39 in total, and that's about 17 of them. You'll see some elderberry in there, a lot of willow in there. These were two examples of things that were in the restoration, elderberry and willow. Part of the not restoration was this snowberry, I think, is Symphyocarpus albus, and this aster, California aster, Symphyotrichium, maybe chilense. These are commonly planted, and these are nice shrubs to invite birds. There were non-natives out there, and there were more non-natives than natives, but again, I think the null hypothesis is like 99% weeds. So the fact that we got 45% natives was pretty amazing. It means the birds are being very particular. You were asking earlier about in making the nest. Not so much, maybe. But in what they're choosing to eat, you have to actively look for those 45%. Of course, the restored plants are often berries, and we don't have a lot of weedy berries. There's some Himalayan blackberry out there as an exception. A lot of these non-natives, like I mentioned earlier, are the mustards. We got all sorts of brassica. That's that Capsella bursipastoralis, the shepherd's purse is the second one there. Lots of radish. If there's a weedy mustard, we picked it up in bird poop. Curiously, and I'm hoping some bird people can help me out here, we found a lot of poison hemlock DNA in bird poop. And I asked a couple bird people that I work with, Alex, for example, who's an expert, and nobody knew. I said, is poison hemlock poisonous to birds? And the assumption is that it's poisonous to all vertebrates. We couldn't find any references for it, except it definitely poisons livestock. So we might be onto something there that might be curious. Of course, we don't want to plant more poison hemlock. There's plenty of it out there. But there might be just a small little novelty discovery there that birds can eat poison hemlock. And we don't know what portions they're eating. They may not be eating the part that Socrates drank the liquid from and was executed from. They may be using the seeds, which might have different compounds.
And that's related to cow parsnip, which is native.
Exactly. However, no cow parsnip out there. And we're 100% sure that's like 100% match. But good question. Yes, that's the weed that you see in most open fields in California. Here's some plant names if you like to geek out on plant names. There's a willow that was both native and part of the restoration, and we hit it 7 times. That's a lot of willows. I don't think the birds are eating the tree. I think they're probably dining on the flowers because they were collected early on, and we know they like to perch there and nibble on the pussy willows, for example. The buds and the flowers. We have some oaks and some blueberries. They like walnuts. All these things have like flowers. I think the oaks, they're not eating, these aren't woodpeckers, they're not eating acorns. They're probably eating the catkins, those little wind-pollinated flowers. Walnut, again, catkins, wind-pollinated flowers. And here's the 3 that were not part of our restoration, the snowberry and the aster, and some ribes. So we're going to start introducing those as part of the restoration in the future. And there's a list of a bunch of weedy radishes and some hemlock and some nasty grasses that the birds seem to also dine on freely. Okay, there's a couple pictures of some things. By the way, that analysis that I just showed you was of about 37 samples. We now have over 100. And we found cute little things like Crasula conata, which is like one of the smallest relatives of Dudleya that fills the edge of salty spaces like the San Francisco Bay. Helenium puberlium makes edible seeds. You might consider that if you want to invite birds around the San Francisco Bay margin. And I talked briefly about the hemlock and the other things. So need to thank a couple people and the people who have funded us. We got money from the Open Space and from the Native Plant Societies. Thank you guys very much. And all sorts of people who we've been working with. But at this point, if there's any time for questions, I can answer questions. Or if you guys have to leave, I know it's getting kind of late. Thank you. Yeah, question up
How do you get rid of the weeds and keep them from resprouting?
How do we get rid of the weeds and keep them from reshotting? I heard that the California Native Plant Society, San Francisco, Yerba Buena chapter is good at pulling weeds. Not everybody wants to pull weeds was the quote, right?
By hand.
But groups like this is where a lot of that happens. Of course, there are also paid positions in weed pulling, and many of my students have gone on to those. They work for places like the Mid-Peninsula Open Space, San Francisco Bay Bird Observatory. I wish I had an easy answer.
Okay.
Heavy labor.
Thank you.
Good question. Others? Yeah, in the back.
So it really does seem like the birds are taking any old thing that's out there. Does that mean that you're pretty confident that the species that you're finding in the bird net in the ancient bird nests represent pretty much everything that was there?
Yeah, we're talking about the bird's nest and whether or not we're getting an accurate description of the community. And I can't say that. I can only say that we have an accurate description of what the birds chose. However, if we go from like not knowing anything about the past versus a snapshot in that perspective, then I think we have these like little Polaroids is what I think about them as. Little Polaroids, and in this one spot it might have looked like this, and this one spot it might have looked like this. And what we're trying to do is generate a plant list for which we can inform restoration practices. And we do know things that could be on that list. The density, like the frequency of them, I don't think is safe because there's lots of reasons from the birds' choices to the DNA degrading at different rates and then amplifying at different rates and our ability to detect it at different rates. So I don't think we could say anything too much about the quantity of those things. But if we detected it, then I think it was there in the 1880s, 1890s. Yeah, there's two perspectives, you know. Do we know everything about it or are we just getting glimpses? And I'm definitely on the glimpses side. It's an important question though. Others, yeah?
Do you have any more bird candidates other than those two in the transitional zone?
Yeah, and there I defer to Alex, and he was pretty sure about those two, but only sure about those two. If anybody knows of any others, I'm all ears. But we're also trying to expand this and think about maybe other ways that we can leverage all of these. There's thousands of these birds' nests in these museums that often are struggling for funding, and we'd like to use them to answer questions in important fields of biology.
Red-winged blackbird.
Red-winged blackbirds, yes, they do like the edges of the San Francisco Bay. Yeah, if you can think of any others, we'll listen, yeah.
I was curious as to the pros and cons of the two different DNA sequencing methods.
Oh yeah, we use Sanger sequencing. That's a traditional approach that was invented in the 1970s. That one has to be homogeneous. You have to have only one thing in there, like one species of plant in your tube, and only worked like less than 25% of the time. But it's really easy, and it's relatively cheap and fast. So that was like our pilot. The next generation sequencing, which is that little harmonica thing I was sending around, you can have mixed collection. So for example, all that environmental DNA of cantaloupe and stuff, we never picked up in the initial study because it's just probably small bits of it. Once it gets running, it's cheaper, but you have to do a little bit of investment in the beginning. and I'm much more confident in the results. So I think, yeah, I think that's the way of the future. My dream is that we go to the museum and maybe with some targeted sampling, but maybe with enough funding, we just sequence as much of the nests as possible. And eventually we're gonna bump into something new to California and hopefully make some insights about what California looked like in the 1880s. It doesn't have to be just a transitional habitat. We could reconstruct large portions of the California flora from 150 years ago with enough support. We have the pipeline for it. So yeah, with the next-gen sequencing, that's how I do it. Good question.
Have you had any conversations with any indigenous people around, see what they have in their
Yeah, that's an important source of information. I cited this study about some residents in Alvisa, and I gotta be honest, those are people who look like me, who were living in Alviso in the 1860s. And there have been people here for thousands of years living there. I have a student right now in my plant class who got an exceptional waiver for the prerequisites because she's a history major, and my plant class is only for biology students. And she's our first indigenous scholarship student at Santa Clara University. And she knows more about plants than I do, which is wicked cool. I don't have that information yet, but I think I have avenues to Ohlone tribes in the South Bay, and I'm hoping they can help out. Sometimes the names are hard for us to reconcile because they know them much more intimately and in a different way than the way I know them. And you could see that here, right? When I started rattling off Phragmites, and I was only communicating with part of the audience. Now imagine like some of the audience speaks a completely different language and has different ways of identifying those plants. But if you know of any ways to connect with those people, I'm all ears.
Well, I'll check in with some Columbia Airlines has competing with some people from around.
Local indigenous people? Yeah. Yeah, I think we have a lot to learn from them in so many ways, but it could be helpful here too. They spend a lot of time, and I've talked to my student quite a bit and her family, in the wetland itself and the importance of those reeds for making boats and for making baskets. And I haven't gotten a lot of information about this really specific spot, this transitional habitat, but part of that is just because it might have had a different meaning to them than the way that I know it. Well, good questions. Thank you guys for bringing so much energy to this big room with the big energy and listening to our story. If you have any more questions, I can answer them up here. If you want to see one of the rarest plants in Santa Clara County, I know I'm in the wrong county, one of the rarest plants in the peninsula-ish, that's the Silicon Valley, or the Live Forever. And I have a mystery nest up here in case anybody wants to see another nest that's not one we've sequenced, but it's filled with lichens and mosses.
Pretty sure it's a bush tip.
And it's probably, some experts say they're pretty sure it's a bush tip. Oh, it does have a flap, we noticed.
Oh, it has the flap.
Yeah, is that a bush tit?
Yeah, it looks like a bush.
Yeah, the bush
It's hanging.
They have like a second door? And part of it fell out of a tree.